Hello and welcome back to everyone to my channel. My name is Shirsha and in this particular lecture we are going to be talking about mammalian cell culture. So in our previous lecture series we talked about protein purification, the different techniques of doing so, what are the advantages and disadvantages of the various methods of cell lysis and chromatography and so on and so forth. Now, protein purification is one set of experiments you would typically be doing in your laboratory work if you are working with the structural aspect of things. If you are interested in structural determination, then you need to purify a protein. But at the same time, just like it is important to know the structure of any protein, that is the uh, arrangement of atoms in a molecule, it is also equally important to know about its functionality in living systems. So, cell culture or mammalian cell culture is a common way or is a very commonly used way to achieve that, whereby for your protein of interest, you can do functional studies in a mammalian system. Why is this important? Because whatever protein you're purifying and whatever subsequent experiment you do with it, may it be some interaction studies or just structural determination or some other physicochemical property characterization, you need to be able to correlate it with its function. Because unless and until you can correlate it with its function and you know a protein's functional importance or what function it plays, it will help you better understand the biology of things. So, mammalian cell culture is a common aim towards that, and it is commonly practiced in many laboratories. So, I'm going to be talking about the different equipments that we typically use in cell culture and the various basics of cell culture, mammalian cell culture. So without any further ado, let us dive right into it. I would like to say, because this topic might seem a little boring at time because it is very informative and there isn't really too much of explanation involved, I will be breaking it up into smaller lectures so as to help you with it. So, mammalian cell culture. So what exactly do we mean by cell culture? Tissue culture is a generic term that encompasses organ culture, wherein a fragment of tissue or an embryonic organ is explanted to retain tissue architecture. What do we mean by explanted? That is, it is taken out from its native location and grown in a culture system. So that is what we mean by explant. So everyone must have heard of the word implant, where we take something from outside and put it into the body system. So explant is just the opposite of it, where you take something from the body system and put it outside or grow it outside. So this would lead to an organ culture wherein the overall tissue architecture is maintained, and there is also cell culture where the tissue is dispersed, that is separated either mechanically or enzymatically, and is propagated as either a cell suspension or an attached monolayer. So you can have both organ culture as well as cell culture. In cell culture, you will be having typically cells that are separated from each other, so therefore no tissue architecture would be retained, and also typically you will be culturing cells of a particular type and not a mixture of different types of cells. Whereas in organ culture, because you will be maintaining the tissue architecture, you will be culturing different types of cells at the same time. Now, the primary determinant in selecting a tissue or cell line for further study is the nature of the observation that has to be carried out. It depends on what kind of studies are you actually aiming to do. Cell culture refers to the removal of cells from an animal or plant, that is a eukaryotic system, or it can be from insects as well, and their subsequent growth in a favorable artificial environment. So just like when culturing bacteria, you have to provide it with a specific medium to provide it with the required nutrients for growth. Similarly, for animal or plant cell culture, you also have to provide a favorable growth medium which will provide it with the necessary nutrients, and not only the nutrients but other components that it requires for its growth like hormones and other minerals and factors. Now the cells may be removed from the tissue either directly, that is, you can simply cut out the cells from the tissue and disaggregated by enzymatic or mechanical means before cultivation. That is, you simply cut out a little bit of the tissue from the system and you separate out the cells either by using some enzymes or by mechanical means. But over here, mechanical means would lead to more disruption of the cells and it might affect the viability of the cells. So enzymatic disaggregation would be better, or it would be more favorable, or they may be derived from a cell line or cell strain that has already been established. So either you can try to establish your own cell line, wherein you have to begin from the tissue from where you would create the explant and take it out and separate out the cells and then culture it. Or you can simply buy a particular cell line from one of the various commercially available companies, or you can just borrow it from some another lab. So basically you're not really creating or generating a cell line or cell strain, you're simply using one which has already been established, for which all characterization has already been done. So there are again advantages and disadvantages of each of these, but we will not be discussing about that over here. We will talk about it later. Now the conditions of growth vary for each cell type, obviously, but generally, so some of the common requirements include a suitable vessel with a substrate or medium that supplies the essential nutrients like amino acids, carbohydrates, vitamins and minerals, growth factors, hormones and gases, that is carbon dioxide and oxygen, and also regulates the physiochemical environment. There is a pH, the osmotic pressure and the temperature. So you have to keep in mind that unlike bacterial cell culture, animal cell culture is a much more complicated and a much more sophisticated system. So therefore, all of your requirements are also more, or you have greater requirements for maintaining animal cell culture. You need to be able to provide media that has all of these essential nutrients, which is the same in case of bacterial cell culture as well. But in addition to the basic nutrients, you need to provide growth factors, hormones, and also regulate the physiochemical environment. That is, not only do you have to regulate the temperature, but also appropriate pH and osmotic pressure. Now, all of these conditions go to ensure that the cells are in an environment as closely mimicking its natural environment so that they can grow to their fullest potential and that they are not under any stress. General cellular processes such as DNA synthesis, membrane permeability, or determination of cytotoxicity may be feasible with any cell type. So if you want to study some generic process, then you can use any of the commercially available cell types for that purpose. While the study of a specialized properties like antibody production or regulation of enzymes involved in particular metabolic cycles will require cell types expressing these specific functions. So what cell type you're going to select for your work will depend on the nature of your work. If it's some generic pathway that you want to study or some generic process that you want to study, then you can use a variety of different cell types for that. But whereas if you want to study any specialized property like antibody production, then you would be needing the B cells for it, or more to be more precise, you will be requiring the hybridoma cells for it. So what are hybridoma cells? Hybridoma cells are nothing but immortalized B cells which can just keep on producing your antibody of interest, and they are immortal in nature. So they will just keep on dividing. They will not die. Or you want to study the regulation of some enzymes involved in a specific pathway. So you will be requiring cells that produce these enzymes that exhibit this pathway, and only then can you study. Or say for instance, you want to study the expression of a specific kind of protein which is functional or which is typically expressed in the body in only a specific kind of cells. So instead of selecting a generic cell line, say for instance, if your protein whose functionality you want to study is expressed in epithelial cells, then you would prefer to select an cell line which is of epithelial origin to do those studies. How you will not be selecting a cell line which is of a different origin, or like for example embryonic origin, for doing those studies. So this kind of considerations you have to keep in mind when selecting the cell line. That is, what studies do you actually want to do with it. So now let us discuss about finite versus continuous cell line. Normal cells usually divide only a limited number of times before losing their ability to proliferate further. Now this is something that you must already be aware of, but if not, typically there is only a certain number of times that a cell can divide before it loses the ability to divide further and it dies. So these cells would be referred to as finite cells. So this is a genetically determined event known as senescence, and such cell lines are known as finite. Now why do typically cells die after they divide a limited or fixed number of times? Because if you have studied molecular biology, and to be more precise, DNA replication and cell cycle and cell division, then you must have read that with every cell cycle, as the DNA replicates, there is shortening of the ends of the chromosomes. And this event shortening can only go on up to a certain extent, beyond which, if your chromosomes shorten further from their ends, then the cells are signaled to die. That is, they undergo senescence. So this is an inbuilt inherent mechanism within the cells to prevent immortality. That is, all cells eventually die. No cells are immortal. So such cells are known as finite cells. Cell lines of a finite life are usually diploid. That is, their nuclei is 2n of 2n status. What do I mean by 2n? That is, they have these two sets of chromosomes present. Again, it might seem a bit confusing to people who are new to biology. What do I mean by diploid? So diploid organisms essentially have two sets of a whole number of chromosomes. So basically you have a whole number of chromosomes that encode for every possible gene in your body. So that is present in two sets. So that is what makes us diploid in nature. So we get one from the mother and one from the father. And so we are diploid organisms. Okay. And these finite cells maintain some degree of differentiation. What do I mean by differentiation? That is, they have some specialization with respect to their structural organization and also with respect to their function. However, some cell lines become immortal through a process called transformation, which can occur either spontaneously or can be chemically or virally induced. So a classical example of transformation that occurs spontaneously is in case of cancerous cells, where we know that cancerous cells can keep on dividing indefinitely and they are immortal in nature. So that is one spontaneous way of becoming immortal, or it can be induced either by using specific chemicals or by certain viruses. Now, when a finite cell line undergoes transformation and acquires the ability to divide indefinitely, it becomes a continuous cell line. Continuous immortalized cell lines are comprised of a single cell type that can be serially propagated in culture, either for a limited number of cell divisions, approximately 30, or otherwise indefinitely. So typically, if you are working on some generalized aspects and you are not looking into very specific functions of specific cell types, in your cell culture, you would be utilizing these continuous immortalized cell lines. And although these cell lines are immortal and they can go on dividing endlessly, typically you would see in practice that you really do not continue a cell line for an indefinite period of time. Because with every passage, with every subculturing, that is with every division, the older the cells get, their functionality gets somehow hampered, and you would see that your experimental results are getting affected as a result of that. So typically we use cells up to a certain passage, and after that we simply go back to the earlier passages and start using those cells again. How do we go back to the earlier passages? We preserve cells in the earlier passages and we carry forward only a small amount of those preserved cells, because with every cycle your cells are going to expand. So to begin with, if you had, say for example, 100 cell culture plates or cell culture flasks, Okay, 100 is too much. Let's say 10. You preserve 9 of them and you have only one which you keep on using. You keep on propagating it until a certain number of cycles, at the end of which you discard it and you go back to that initial uh, passage number, or to the initial phase, that is the 9 that you had stored, and you take one out from there and you go ahead with it. So this is how you use the immortalized cell lines. Although they're immortal, you do not use them indefinitely to ensure that the cells are in their optimum health. Some common examples are HeLa, which is a human cervical carcinoma derived cell line. We have the HEP2, which is a human epithelial cell line, and BHK21, which is cells derived from the kidney of a baby hamster. Continuous cell lines are subcultured indefinitely, in theory, in glass or plastic surfaces or suspensions, as they are usually obtained from carcinogenic cells. So you can have either suspension cell cultures, or if you have adherent cell cultures, that is cells that require a surface to attach to, then you will be using these glass or plastic surfaces. Now typically, um, a lot of the mammalian cells that you culture are adherent in nature, because naturally they would be found in the extracellular matrix embedded in the ECM in the body. So this adherence is essential for their survival. So most cells are adherent, but some cells, for example the blood cell lines or certain specific modified cell lines, can be grown in suspension. These cell lines are used most often because of their faster and longer growth, less nutritional requirement, and high plating efficiency. So now we are going to be talking about the various different equipments that are used in mammalian cell culture. So to begin with, I have already given you an introduction about cell culture. That is what exactly it is that we mean by cell culture, and the two different kinds of cells that are commonly used in cell culture, that is the finite cells and the continuous cell, and how you can decide which specific cell line to select for your particular work. Now mostly typically you would be working with continuous cell lines unless your work requires you to work with finite cells, or for instance if your work requires you to handle terminally differentiated cells like neurons. So because neurons are already terminally differentiated and they will not divide any further, so you would every time need to prepare a fresh cells from an explant. So in that situation, you cannot use a continuous cell line, but mostly for most of the work, you can use continuous cell lines. Obviously of specific origin, depending on what kind of studies you want to do. That is whether the system that you want to study is naturally observed in the epithelial cells or endothelial cells. So accordingly, you will select your continuous cell line of specific origin. So now coming to the various equipments that are used in mammalian cell culture. So first and foremost, we have the biological safety cabinet. So the biological safety cabinet, or BSCs, is like your laminar hood. It's an upgraded or more sophisticated version of a laminar hood. Air is filtered through HEPA filters, which are high efficiency particulate air filters, before passing across the workspace, and again before it is released outside the cabinet. So there is a dual degree of protection. You want to first filter the air that is going to pass over your workspace where you have your cells and various other things that you're working with, because you want to protect them from the contaminants that would be present outside in the environment. And the second level of filtration is before you release the air outside the cabinet, because it might be quite possible that you're working with something which is toxic for the environment. Under such circumstances, you have to filter the air before you can release it out to the environment, because you always have to be a responsible user. You cannot release any toxic substance out into the environment. So filtering the air both before it enters the workspace and before it exits the cabinet are equally important. Now, biological safety cabinets come in several classes, 1, 2, and 3, that offer varying levels of protection from hazardous or contagious materials. HEPA filters remove at least 99.97% of airborne particles that are greater than equal to zero point 0.3 micron in diameter. HEPA filters are composed of a mat of randomly arranged fiberglass fibers with diameters in between 0.5 and 2 micron. The key factors affecting function are the fiber diameter, that is how thick the fibers are, the filter thickness, that is how thick the overall filter is, and the phase velocity. Now, what do I mean by phase velocity? It's the velocity at which the air is being made to pump through the filter before it enters the workspace and exits the cabinet. Now the cabinet should be turned on about 10 to 20 minutes before being used because you need to give it appropriate amount of time to establish a specific pressure at which the air will be flowing through the cabinet. The cabinets are typically equipped with a short-wave UV light that can be turned on for a few minutes to sterilize the surface of the cabinet before use. And after your use as well, you should always turn on the UV to sterilize the hood for the next use or for the next person. However, only exposed surfaces will be accessible to the UV light. All surfaces should be wiped with 70% ethanol both before and after each use. So particles passing through HEPA filters are trapped through a combination of the following mechanisms. First is interception. So interception is where particles following a line of flow in the airstream come within one particle radius of a fiber and simply adhere to it. They will get absorbed onto the surface of the fiber. Next, we have impaction, where larger particles are unable to avoid fibers by following the curving contours of the air stream. So you have the fibers and you have the air curving its way through the fibers. So these larger particles, because of their larger size, are unable to curve so much as the air, and it will simply come into, or it will simply go on and hit these fibers, and they would then get embedded in them. So they are forced to embed in one of them directly. This increases with diminishing fiber separation and higher airflow velocity. So less spaced your fibers are, that is closer your fibers are, and faster the velocity with which the air is flowing through it. So the air will rapidly curve and make its way through the cracks and crevices or the spaces in between the fiberglass fibers. But the larger particles, owing to their higher inertia, they will be unable to move so much and they will simply go and bump into these fiberglass fibers and will get embedded in them. So this will filter them out. And the third method is diffusion, which is a result of the collision with the gas molecules by the smallest particles. So the largest of the particles, they will be removed by impaction. Then we have the smaller particles which will be removed by interception. But then there are even smaller particles which would generally be not subject to impaction, and they can avoid interception if they do not come within this specific distance of a fiber. That is, if they do not come within one particle radius of the fiber. So what happens to these particles? As they are moving, they will collide with the gas molecules, right? So those particles whose size is less than 0.1 micrometer in diameter, they will collide with the gas molecules, and this will reduce their speed of migration through the filter. So which will thereby impede and delay them in their path through the filter. So as they're impeded and as their movement through the filter is delayed, it increases the probability that a particle will be stopped by either of these two above mechanisms, that is either interception or impaction. Okay, so this typically becomes dominant at lower airflow velocities where it's moving slowly and it's colliding with the gas molecules, and now this collision will make its movement even slower, increasing the chances of it being removed by either interception or impaction. Most likely interception and not impaction. So this is what the cross section of a typical HEPA filter looks like. So you have this continuous sheet of the filter media, and this is your frame holding it. So this is how it looks. If you look or inspect it closely, you have these randomly arranged fiberglass fibers. So interception is when you see this blue line, which is marking your pathway of airflow. So these particles, while they're flowing through in between these spaces in the fiberglass, when they come within close approximation of the fibers, they will get absorbed onto the surface of it. So that is what we call interception. Impaction is when you have these larger particles, which the air is simply bending and flowing, bending, and it's flowing. What do I call it? It's flowing past these fiberglass fibers. That is, it's simply making its way around the fiber. But this larger particle, because it of its higher inertia, it cannot make its way around the particle, and it will simply come and hit it, and it will get therefore embedded into the fibers. And then you have the even smaller particles, smaller than those that are getting removed by interception. And these particles, because they will be colliding with the gas molecules, and this collision will slow down their movement or rate of migration through the filter, and this will increase its chances of removal by interception. So there are different classes of BSCs, like we've already mentioned, class 1, 2 and 3. So we are going to look at them very briefly, like what is the differences between the three classes. So first we have the class 1 biological safety cabinets. Now these provide both operator and environmental protection, but little product protection. So both the user, that is the operator, and the environment are protected, but the product that you're handling with, or that you're handling inside this hood, or that you're working with inside these safety cabinets, are not really protected much. These BSCs are commonly used to enclose specific equipments like centrifuges or procedures like aerating cultures that potentially generate aerosols. So these aerosols otherwise would be detrimental to both the operator as well as to the environment if released. So you will be typically using a class 1 BSC when dealing with such cultures, which produce these aerosols which can be harmful. But because these cultures don't really need to be protected from the environment as such, so therefore you can use these class 1 safety cabinets for them. So this is what a typical class 1 biological safety cabinet looks like. If we take a look at the pathway of airflow, you have this unfiltered air which is entering within the workspace of the safety cabinet, and this is a room air making its way over here in making its way into the workspace directly. There is no filter over here, but because the air is flowing from outside to inside, because your user will be sitting over here, so the user is protected. It is flowing from outside to inside, so it's flowing away from the user, and nothing from within the workspace is getting carried towards the user because the airflow pathway is like this. And then you have the HEPA filtered air over here, you have the filter here, and whatever air is being thrown out back to the environment, that is again filtered out. So both the operator and the environment are protected, but the product not so much. Next we have the class 2 biological safety cabinets. So these provide appreciable operator as well as product protection. A curtain of sterile air is drawn over the products that are being handled. The air is then drawn underneath the work surface and back up to the top of the cabinet, where it passes through the HEPA filters. Part of the air being drawn into the front of the cabinet underneath the work surface is exhausted, and the rest is recycled. The air being drawn in acts as a barrier to potentially contaminated air coming back out to the operator. So what happens if we take a look at the pathway of airflow for a class 2 biological safety cabinet? You have this curtain of air that is created. So the operator or user would be sitting somewhere over here. So air is definitely being drawn in from outside, but unlike in case of class 1 BSCs where the air was simply filling up the entire workspace, in class 2 BSc, this air does not fill up the entire workspace, but rather it gets drawn into this space underneath the workspace. So there is this empty space underneath the work surface, and over there the air gets drawn in, and then it is getting pumped up and it is being filtered. And after it is filtered, some amount of it is being released back to the environment, and the rest of it, or the majority of it, is being pushed down onto the workflow. Now, as it is being pushed down onto the workspace, so basically a curtain of air is created. Where is this curtain created exactly? So it is created over here, in between the user and the cell culture, that is, or the product that is being handled. So this air, it might look like it's falling down on the entire workspace. It is filtered air coming down here. So it will create this curtain between the user and the product. So both the user is being protected from whatever might come out from here, and the product is being protected from the outside potentially contaminated air. And wherever the user simply puts in their arms into the laminar hood, that is where the curtain would get disrupted. So this is what a typical class 2 biological safety cabinet looks like. So you see over here, so you would typically be working with these class 2 BSCs in your laboratories. So you would see that at the very front, you will see some empty space on the work surface. If this is a stainless steel work surface, you will see that there are some perforations at the very beginning. That is the place through which your air is getting passed into the space underneath it. And this surface is typically removable. You can lift it up and you see that there is a space underneath it. So this is the space where the air is being drawn in, and this actually completes the entire cycle. So it's getting drawn in from here, it's getting pumped back up. This is where you have the filters. Some air is being exhausted out, and the rest of, or majority of it, is being pumped down over here onto the surface to create the sterile curtain of air. Now, class 2 BSCs can be of again different kinds, and this classification depends on what is the velocity at which the air is being pumped through the safety cabinet, and also what is the proportion of air that is being exhausted and that is being recycled back and released onto the laminar hood workspace. So depending on these, there are different classifications, but we are not going to be discussing about that over here. Finally, we have the class 3 biological safety cabinets, which provide the utmost or the highest degree of protection. So class 3 biological safety cabinets are specifically designed for work with BSL, that is biological safety level 4 pathogenic agents. It provides maximum protection to both the user and the environment and the product, and is generally installed only in maximum containment laboratories. Now, if you think of the laboratories at NIH that are working with the COVID virus, so they would definitely be using class 3 biological safety cabinets for that. And even in your science fiction movies, or those movies where you have scientists who venture out into space to collect some alien life and they're studying with it, the hoods, the laminar hoods that are typically shown, those are actually nothing but class 3 BSCs. So this is what it typically looks like. I'm pretty sure most of you are familiar with it. If not from an academic point of view, then definitely from a movie-based point of view. So you have these holes wherein you put in your hand, and there are these hand-shaped gloves within. So nothing from inside comes outside, nothing from outside is going inside. Whatever you're working with stays inside, and there is this special cabinet on the side through which you can put your sample in, again in a very protected and in a very contained fashion. And unlike in class 2 biological safety cabinets, where the curtain of air was being disrupted by your arms that you are putting into the laminar hood to work with, over here there is no such disruption, no risks. You will not even be directly touching the items that you're working with. So this is how the pathway of airflow looks like for a class 3 biological safety cabinet. So this is where you put in your hand through these glove, and this is where the air is getting recycled. So you have unfiltered air under negative pressure, it's getting pumped up, and room air from the top is being directly filtered in. So you're going to work in this front part and not at the very back, because at the very back you would have some unfiltered air, but at the front, that's where you're going to work with the things. Well, I'm sorry, yeah, that's where you're going to work with your, or that's where you're going to handle your BSL-4 pathogenic agents. Also, unlike in case of class 2 biological safety cabinets, because the air was getting drawn in from outside, so therefore it was going through from below the workspace and getting pumped up. But over here, since the air is being pumped in directly from the top, so it has to be circulated within this space itself. There is no way that you can make it pass through elsewhere. Okay, so typically there would be some perforations on the back wall, and the air pressure would be such that these unfiltered air under negative pressure would be in that back space where you would not be able to even reach. So there is no risk of contaminating your samples. So in case you are interested on learning more about biological safety cabinets and how exactly the air pressure mechanism works, if it was not really clear to you and you want to read further on it, or you want to learn about the different subtypes of class 2 biological safety cabinets, then you can visit this link. This has a very comprehensive take on, or a comprehensive explanation of the various different kinds of biological safety cabinets and their mechanism, or the mechanism of their functioning. So next we are going to be talking about another very important equipment that is required for mammalian cell culture, and that is your CO2 incubator. So the purpose of the incubator is to provide the appropriate environment for cell growth. That is, it will maintain the appropriate temperature, the appropriate level of gases, that is CO2 and oxygen, as well as the appropriate level of humidity. CO2 incubators are important in the cultivation of mammalian cells, where the relative humidity is typically more than 95% and a slightly acidic pH is achieved by maintaining a CO2 level of 5%. So you would typically have CO2 cylinders that are linked up to your CO2 incubator. Insulation is maintained by either a water jacket or an air jacket with corresponding advantages and disadvantages. The water jacketed incubator can maintain temperature over a longer period of time should there be a power failure, because as we know, water would provide much greater insulation as compared to air, and therefore if there is a power failure and your backup generators do not really kick in that case the water jacket will help maintaining the internal temperature for longer periods of time. It is, however, much heavier when filled, and the level must be maintained by periodic topping off, because the water will keep getting evaporated and the level will go down. So you have to maintain the level of water by periodically refilling it. In contrast, the air-jacketed incubator is much lighter. It comes up to temperature faster because, unlike in case of the water-jacketed incubator, over there you have to give time for the water surrounding the incubator to get heated up, and only then can it maintain the temperature within the incubator. So in contrast to that, the air jacketed incubators come up to temperature much faster, but will lose heat much faster when the fan goes off, as in case of a power failure. Now, culture flasks should have loosened or filter caps to allow for sufficient gas exchange. Typically, we don't use loosened flasks, but the flasks that loosened caps, but the flasks that we use for cell culture have filters already fitted in the cap. So you can just tightly secure the cap, and the filter will allow for sufficient aeration or exchange of gases. Cells should be left out of the incubator for as little time as possible, and the incubator doors should not be kept open for very long, because in that case the CO2 level would go down, the temperature would also be affected, and in fact the humidity within the incubator would also be affected because it would be lost to the outside environment. For supplying CO2, commercial gas cylinders are used, not maybe, but commercial gas cylinders are used. And the critical point is to maintain an uninterrupted flow of gas. So typically, if you have animal cell culture set up in your laboratory, you would see that in addition to the CO2 tank, which is hooked up to the CO2 incubator, you have additional backup cylinders somewhere close by, so that in case your CO2 runs low, you can simply replace it with the backup cylinder, because it is very important to maintain CO2 levels in the incubator, and if there is any disruption, then your cells will begin to die. And obviously, because these are commercially available cylinders, the inlet through which CO2 is entering into the incubator has a filter attached to it, through which the air will get filtered as it enters inside the incubator. So a miniature regulator is used to reduce the gas to a flow rate optimal for the incubator. You can't just let it rush in. So you can regulate the pressure at which the gas is being released from the cylinder and is entering the incubator. And an inline 0.2 micron filter. So what do I mean by inline? There is a filter that is fitted in the path of the airflow, that is at the tip of the pipe. So if you have a pipe coming from the cylinder towards your incubator, then the mouth of the pipe will have the filter. So that is your inline filter, which prevents introduction of mold and other potential contaminants from the CO2 itself. So this is what a typical CO2 incubator looks from the outside. You have the display which shows the temperature, and then you have this other display which is showing the CO2 level within the incubator. And next you have, and this is what the inside of a CO2 incubator typically looks like. So this is where you have the jacket, and most of the incubators typically have an air jacket, and you have this fan over here that is helping in filtering the, or rather pumping the air inside outside. So it's exhausting the air that is inside outside. And you have this tray underneath which you refill with autoclaved milli-Q water, that is double distilled, double filtered water, which helps in maintaining the humidity. So you have to ensure that this tray is refilled periodically and it doesn't go dry. If the tray goes dry, in that case the humidity within the incubator will be affected. You have this first glass door, so in case you want to check something, then you can simply open the outer door without opening the inner glass door, and because it is transparent, you can just see through it and see where your cultures are kept or how things are looking. If you just want to look at it, have a glance. So this helps in ensuring that the inside carbon dioxide and oxygen levels and humidity and temperature is maintained without dropping it much while you want to simply look at things. So next we have the inverted microscope. So inverted phase contrast microscopes are used for visualizing the cells. An inverted microscope is a microscope with its light source and condenser on the top, above the stage pointing down, while the objectives and the turret are below the stage pointing up. So inverted microscopes are useful for observing living cells that are adhered at the bottom of a large container, for example a tissue culture flask, under more natural conditions than on a glass slide, as is the case with a conventional microscope. So if you have your conventional microscope, because your objective lens would be on top of your specimen, so you have to have your specimen on a flat surface so that you can see it. But imagine you have a petri dish in which you have your cells settled on the bottom of the dish. So the best way to look at it would be using an inverted microscope, where your objective will be below the specimen. So if you have your sample over here, so let's imagine that this gray rectangle represents your petri dish, and at the bottom surface of this petri dish you have your cells. So if you have your objective just below the surface, you can easily see the cells while they are within the petri dish. If you were using a conventional upright microscope, you would not be able to see it, and in that case you would need to transfer the cells onto a glass slide, but then the cells will not be in their natural environment. So therefore in cell cultures typically we use inverted phase contrast microscopes. Why phase contrast microscopes? Because if you are visualizing living cells in their culture dishes, then you will not be staining them. So how do you then see something or see some transparent living cells without staining them? By using phase contrast. That will help differentiate the cells from the surrounding environment. So this is what a typical microscope looks like that you will find in your any typical cell culture laboratory. Mostly we use binocular microscopes, that is they have two holes through which you can look into it, so it makes it easier on the eye. We have the vessels for cell culture. So plasticware, although expensive, is preferred over glass for pipettes, tissue culture dishes, and so forth. Why is plasticware more expensive? Because it is not reusable. It's more of a use and throw kind situation. So that makes it more expensive as compared to glass. But it is again preferred over these glass items because you will not be reusing them, so it reduces the chance of contamination. You will be buying pre-sterilized plasticwares and you will be simply using them directly, so that reduces the risk of contamination. Glassware used for tissue culture, on the other hand, need to be autoclaved with moist heat for 30 minutes at a pressure of 15 pounds per square inch. Anchorage dependent cells or adherent cells, that is most mammalian cells, require a non-toxic, biologically inert and optically transparent surface that will allow the cells to attach and allow movement for growth. So adherent cells require this surface on which they can attach because this adhesion is essential for their survival and for their growth and replication. Now, why do you need this surface to be optically transparent? So that you can visualize the cells using the inverted microscope. If it was not transparent, then you couldn't see the cells. The glassware would be blocking your view. It has to be biologically inert because the surface by itself should not in any way affect the growth of the cells. So it has to be non-toxic and biologically inert. The most convenient vessels are specially treated polystyrene plastics that are supplied sterile and are disposable. So these are the most convenient. These include petri dishes, multi-well plates, microtiter plates, roller bottles, and also the screw cap flasks. Suspension cultures can be shaken, stirred, and maintained in shaker flasks or grown in vessels identical to those used for anchorage-dependent cells. But typically, you will not be growing them in vessels used for the anchorage-dependent cells, and you would preferentially use specific flasks that will be shaken or stirred to ensure that the cells do not settle down at the bottom. So this is typically some example of some glassware. So these are called the various cell culture flasks. These are used to grow adherent cells and they come in different sizes, demarcated by either T25, T50, which represents the surface area that is available. That is the number 25 represents the surface area available to the cells for growth. Similarly, 50 or 75 would represent the surface area available to the cells for growth. Then you have these multi-well plates, you have the 24-well plates, the 6-well plates, and different other multi-well plates, which are also pre-treated. So if you are going to grow adherent cells in them, you typically do not need to subject them to any treatment. You can just directly use them. But sometimes there are some multi-well plates available which require some coating or pre-treatment to be done to them before they can be used for growing adherent cells. So this coating typically involves either a coating with 0.01% polylysine or with collagen to ensure that the cells indeed stick and take on the appropriate morphology. And then these are the flasks that are used for maintaining suspension cell cultures. Lastly, we have the liquid nitrogen freezers. Now, these are as important as any of the other equipments that we have talked about. The other equipments that we talked about so far were required for maintaining the cells, for letting the cells grow, for visualizing the cells, and so on and so forth. But the liquid nitrogen freezers form a very integral part. They help you to preserve your cells, to store your cells. Like at the beginning of this lecture, I told you about how it is important to preserve your cells in the initial levels of passage, because typically, although continuous cell lines are theoretically immortalized, so they can be used indefinitely or they can be grown and cultured indefinitely, but you would preferentially not do so. So You must store cells in the earlier passage to have something to go back to. And where do you store these cells? In liquid nitrogen freezers. So liquid nitrogen freezers are for long-term storage, and these are necessary for any tissue culture facility. They are usually portable units which can be moved as and when required. It is necessary to regularly monitor the liquid nitrogen levels within them because you do not want the level to fall below a certain mark. So liquid nitrogen is a very good preservative agent. It helps maintain a very low temperature of minus 196 degrees centigrade, at which your cells would be metabolically dormant. So this is what typical liquid nitrogen tanks look like. They come in different sizes and they are fitted with wheels at the bottom which make them portable. And within these containers, you basically have slots made wherein you can hang in racks. And these racks again have slots where you can store liquid nitrogen suitable boxes. So you can't just store any box. You have these special boxes available which will not be damaged when kept or stored in liquid nitrogen. So you have these racks within these tanks where you're storing the boxes. So you can imagine, like, let me help you visualize it. So you have a rack like this over here going down, and then you have these slots on the rack, and then on these slots you have your boxes. So in the slots you put in your boxes, and then you put in this rack with the boxes within the nitrogen tank. So you have like a lot of racks in one tank, and the number of racks that a particular tank can house will depend upon the size of the tank itself. So it is important to periodically refill the liquid nitrogen level, because if your level goes down, say for instance still here, then your topmost box is getting exposed. So that is not good for the cells. And this is what a typical this is again like how do I call it? It's used for storing cells in minus 80 degrees centigrade freezers. So when you're making freeze downs of the cells, you would not directly keep them in liquid nitrogen, but rather you will first keep them in minus 80 for overnight, and then the next day you will be shifting them to your liquid nitrogen tanks. Because if you shift the cells directly to liquid nitrogen tanks, then that will be detrimental for the cells again. So you put them in these special boxes. Now, if you keep the cells directly at minus 80, it will cool very rapidly, and there is a risk of ice crystal formation that might disrupt your cells and kill your cells. So what you do is that in these special containers, you can see that there are these slots for putting in the vials containing your cells in the specific freeze down media. We will discuss about that in later lectures, what it is and how it is made. And this space is actually meant to be filled up with the fluid, typically isopropanol. So because that would freeze very slowly, and therefore will allow the temperature within the vial, that is the cell, to freeze down slowly. It will slow down the rate of freezing and therefore impede the formation of ice crystals. And once your cells have frozen overnight, the next day you take them and transfer them to the liquid nitrogen. Now, you can store your cells at minus 80 degrees centigrade, but they won't be viable for very long. Maybe for a couple of months, it's fine. But if you want to go for long-term storage, where you're storing your cells over a period of years, then storing them in liquid nitrogen tanks is the best option. So that is all about the various equipments that are required for mammalian cell culture. So to briefly recapitulate, we talked about the biological safety cabinets, which is the laminar hood where you work with your cells. We talked about the different classes, that is class 1, 2 and 3. And we also talked about the mechanism of airflow, or the pathway of airflow in these different classes of biological safety cabinets, and how it offers varying degrees of protection to the operator, to the sample and to the environment. Next, we talked about CO2 incubators, which are again very essential for providing the appropriate environment for growth. That is, they help maintain the temperature, the CO2 level, the pH, the humidity. So the temperature, CO2 level and humidity, these three factors are maintained by the CO2 incubator. For maintaining pH, that is, well, the CO2 incubator also plays a role in maintaining pH, but we will not discuss about that over here right now. We will talk about it in the next lecture. Then we talked about the inverted phase microscope that we use for visualizing the cells, and that is for periodic monitoring of the cells, how they're looking morphologically, if they're healthy or not, if there is any contamination or not. Next, we talked about the various vessels that are used for cell culture, the different glasswares that are used, and finally, we talked about the liquid nitrogen freezers. So that is it for this particular lecture. In the next part, we will be talking about the different kinds of media that are used for mammalian cell culture. What are the components of the media? What is the utility of each component? And so on and so forth. How pH is maintained and all of those things. So I'll catch you next time, and until then, bye. And in case you have any questions, feel free to leave it in the comment section. I will definitely get back to you. And if you like this video, do share it and subscribe for more such content. So yeah, coming back to it, bye.